We were talking about basidiomycetes last time. So basidiomycetes are, like I said, they're probably the ones you are most familiar with in the sense that you see them a lot around here when you're hiking with the fruiting bodies or mushrooms. Most of those of what you see are basidiomycetes, not all, but most. And so, and a lot of them are ectomycorrhizal fungi, which I'll talk about in a little bit here. And so they have a kind of a complicated life cycle. And again, I'm not going into the details of the life cycle, but it is this idea, again, of a plus minus mating type of the hyphae meeting party. And it's usually in the cap region that you will see the sexual reproduction going on in discrete areas there in order to form sexual spores that will then be released. So basidiomycetes, here's a close-up of the life cycle. And again, I'm not getting into detail, but just showing the big picture idea. And so when you are foraging for mushrooms, really from their perspective, the reason they did that was to finish off their sexual reproduction. And most fruiting bodies are above ground so that the spores can get dispersed via wind dispersal. You may have encountered something like this at some point where they talk about a fairy ring of mushrooms. And that is often the pattern that you'll see. And again, these are most likely mycorrhizal fungi that are hanging out with the roots of that tree. And you have to appreciate that roots can extend quite a bit from, you see the stem, of course, coming up out of the ground, but the roots really can extend quite a bit. And so it's not all that unusual to see mushrooms popping up at a fair distance, but sometimes they'll be in that cute little fairy ring around the tree. So sometimes I'll have someone bring me a fungus and say, what kind of fungus is this? And if it's just a bunch of hyphae that they gave me, this is what I do. I look at it under the microscope and I look for plant connections. So this is very distinctive for basidiomycete fungi. They form these weird little bridges, and they think that it's a better way of kind of systematically dispersing their nuclei along the length of the hyphae as it's growing. Don't know for sure if that's accurate, but sounds reasonable. But anyways, very characteristic. If I see this under the microscope, I can say with really good certainty, yeah, I think you have some kind of basidiomycete. It would actually look like this under the microscope. So that's very characteristic. Otherwise, it can be hard to tell. The best way to tell what kind of fungi you have is actually, I used to teach a technique where you grow the fungi on agar that's already sitting on a microscope slide, and then go through this little procedure to grow it, and so that it'll grow down onto the slide. And the reason you do that is you don't want to disturb how they form, especially where they're forming spores. And it's so easy to disturb that. So if you get it to grow out onto the microscope slide, you preserve their method of spore production, and then it's much easier to tell what kind of species you're looking at or group that you're looking at. So if you took some gill tissue and looked at it under the microscope, so the underside of a lot of mushroom caps, you see the gills, and so that here you have 3 gills, and the blue is the normal fungal hyphae, and then the brownish looking things, those are the sexual spores that are being produced along the edge of each gill. And so you can get an appreciation. They produce a lot of spores. And this is just 3 gills that are in just, a fraction of what you're really looking at if you look at the whole cap, mushroom cap. So very prolific spore producers. But again, that's partly due to the fact that it's air dispersal. And so they usually go with quantity and hope that some land and make it. And so you can see all kinds of different fungi around here, fruiting bodies. If you hit this past fall was a pretty decent. Did anyone take mycology with Dr. Robertus this fall? I think he offered it right in the fall. Anyways, I'm guessing it was a pretty good year for fungi and hunting for fungi and fruiting bodies. It can be really cute and small as well. if you look underside of the cap and you see pores instead of gills, if you looked at it under the microscope, it would look something like this. So here you see a handful of pores, and you would be zooming into the tissue that's lining the perimeter, I guess, of the pore, and that's where you would find the spores getting produced. So slightly different strategies, but and of course, not all fruiting bodies are on the forest floor. Often you see growing on stem trunk or on trunks or on branches. Lots of different colors and pigments. Puffballs. Don't inhale. So when I go hiking when my boys were toddlers, of course, if they found a puffball, they wanted to poke at it, get the spores. Have any of you done that when you were younger? It's so tempting to poke at it and get the spores up. And so I always make them, I'm like, okay, you can do that, but first, figure out what direction the wind is coming from and be upwind of where the spores are going to fly. There was a case study of teenage high schoolers thinking that if they inhaled these spores, they'd get high. They thought they were hallucinogenic. And so they were at some outdoor party and they all just got that idea in their head, probably after imbibing some alcohol and making poor decisions. And so they decided to poke at it and inhale it. And they actually got really sick, had to go to the hospital, and they had survived all, but not a good idea. That can get into your lungs. There was another case study where an adult didn't purposely inhale, but did get exposed to spores, and they actually started growing in his lung tissue, like the fungal hyphae. So when they open them up, it's like, oh. So yeah, be really careful. They're fun to play with, but don't inhale. And then I also heard another case study of a elderly patient undergoing surgery. This was back in the day, not recently. And the surgical room up in the corner of the ceiling had mold that no one really paid attention to, which I find amazing that they didn't clean the room better than that. But anyways, the spores wafted and ended up in the chest cavity. He was doing open, I don't know if it was open heart surgery, but anyways, his chest cavity was opened up. And he seemed to, he came out of the surgery okay, but then several days later got really sick. And they had to reopen him, and he had fungal hyphae growing in his chest cavity. So that kind of thing is just rather alarming. So you have to be respectful. He did live. They were able, luckily, the fungus did have sensitivity to antibiotics, and they were able to deal with that. But when you look at all the antibiotics out there, there's really not that many that can kill off fungi. Well, let me rephrase that. There are antibiotics that can effectively kill off fungi. The problem is when you take them, they kill you too. It's this, we're too similar. Fungi and humans are too similar in some ways in that regard. And so it's really challenging to find an antibiotic that only targets the fungus and not the human host. So not fun. Anyways, on to better, happier topics. Chanterelle. Does anyone have tasted a chanterelle? Apparently they're quite tasty. I have yet to do that. Someone told me they taste like apricots, which I thought was interesting. So one of these days, I'll have to try to sample that. Very much edible. This one's not. This is Amanita, deadly. Looks very inconspicuous. I've seen these around for many years in this area. So I don't know if any of you have taken a class with Dr. Robertus where he might talk about mushrooms, but he told me a story when his son was in elementary school. The science teacher took them out and they went and foraged for cool looking stuff and made like a collage out of it, stuck things to a poster board, and then they displayed it, and then they had parent teacher conference and everyone could see everybody's poster boards of all the cool things in nature. And Dr. Roberta said, I almost had a heart attack. I went to many of the posters and there was Amanita bits and pieces glued to the poster. And he said, when you consider the body mass index of these kids, these things could have killed them if they had accidents. So kind of alarming. So you got to know what you're doing when you're doing this kind of thing, especially with young kids. This is another one. This is a little bit more flamboyant, where you might go, maybe this one's poisonous, and you would be right. I don't know if it's this one. What is the one? Have you heard that it's an Amanita species that they think allowed the Vikings to go berserk? You know, the berserkers. You heard that story? Dr. Robertus teaches that, talks about that one too, and I can't remember if it's muscaria or a different Amanita. But apparently the Vikings would like cook it up, and I don't know if they drink and eat, but like drink the broth and eat bits and pieces, and then it would allow them to hallucinate and not feel much pain. And then they go into battle. And I think that's how they got the term berserkers, because they were kind of off their head with hallucinating. And when you're in that condition, you don't feel pain. So I guess you'd be a good warrior. So it's one of the Amanita species. I can't remember which one, but again, this one looks pretty, innocent looking, and again, it can kill you. So just be super respectful. I will look at fungi in the wild, but I won't really touch them. Examine them, admire them, but I'm careful around them because I don't know enough about some, especially some of the more innocuous looking ones. Jelly fungus, really cool looking. Moose ears, that's the common name for this one. Moose ears. Bird's nest fungi. So they have this cup-like structure with these pieces of fungal tissue inside. Here's another kind of better image there. Bird's nest. Really amazing designs, really cool. Very dainty. Look at this one. This one doesn't have such a nice name. Veiled stinkhorn. That's the name of that one. So I'm guessing it's stinky. This one looks pretty ordinary. This one's called Armillaria mellia. And when this one was discovered, I was in grad school, and this is the humongous fungus from the Munising area. And so this is the scientific article that was published. Johann Brun is one of the authors, and he was one on my PhD committee. He worked in the forestry department at Michigan Tech University. And so Bulbosa and Mellia have been, they're the same thing. This one was claimed to be the largest and oldest living organism at that time. And since then, there's been others to surpass that, but the Johann, he was kind of a quiet person and not very, I would say he was an introvert and, not very willing to engage with the media. So they would keep calling. When this came out, it was a big deal. And so people from news agencies, were calling and saying, we want to bring our videographer or photographer out. Can you take us to the woods, show us this huge fungus? So that anytime he was interacting with these people, they thought there was like a huge mushroom in the forest that they would get to photograph, take video on it, sensational news piece on the evening news or whatever. And he got so sick and tired of that. And so he started taking, like, he deliberately trudged them through like terrible sections of woods where they're bushwhacking through it, and just really arduous and difficult. And then he'd bring them to, just an ordinary patch. And he said, okay, we're here. And they're like, where's the mushroom? And he said, no, it's all underground. It's all there. We're on top of it, but it's the largest living organism underground. That's not part of the title. So he started to have a little fun with it. He got so frustrated with it, decided to poke fun. But it really is underground. And then when they do come above ground, they're just ordinary looking mushrooms, ordinary size, nothing fancy. But they were able to show that underground, it was all the same organism. It was just basically the same organism that had grown through this area in Munising. And to this day, they still have humongous fungus fest in the Munising area. They took a break during COVID, but they started up again after that. And so the restaurants serve, humongous fungus burgers and all kinds of things going on, including a parade, I think. So lots of local interest on that. Okay, one other group I wanted to talk about. Glomeromycetes. This is relatively new group. So these are fungi that are obligate mutualists of plants. So obligate, that means at least to date, we haven't figured out how to grow them separate from the host plants. It doesn't work. And so we call them obligate to their plant host. So we're actually talking about arbuscular mycorrhizae type fungi, and I'll talk a little bit more about those. So they've been called, they are called several different things. Currently, it seems in the published literature, arbuscular mycorrhizae is the favorite way to talk about them, but they're also known as vesicular arbuscular mycorrhizae or VAM fungi, and endomycorrhizae is another term. So all of those mean the same thing. I tend to say VAM fungi because it's nice and short, rather than saying arbuscular mycorrhizae or AM. That doesn't really have much of a ring to it. So I tend to say VAM. So if you hear me saying VAM, I'm talking about these. So they are very, very interesting because they form a very intimate connection with the plant roots and provide some very valuable services to the plant. So I'll talk a little bit more about that in the next slide set. The Glomus species is the largest genus so far discovered in this group. And you can find a lot of Glomus species for sale if you want to add in mycorrhizal fungi to your soil. Often when you look at what is in that package, it's usually varieties of Glomus species. So these are, like I said, relatively new class. This is a 2001 paper, a new fungal phylum, the Glomeromycota. And so here, this is just a little blurb from the abstract. And so again, it's not that long ago that they decided to, when they did enough studies on them, they're like, yeah, they really do belong in their separate phylum and class. They used to be, I think, lumped in with the Astromyces. Or no, I think. So we got them separated out. So the glomeromycetes, when we say they live intimately with their host root, the plant roots, here's some images of that. And so some of the images that you see on here are under the microscope. And what they do is the fungal hyphae gets into the root tissue and they form what we call vesicles. So that's why you get the vesicular term. That's just fungal hyphae that's ballooned up. So it just pops up. Okay, so but that's fungal hyphae. And then the arbuscle idea. So vesicular and now arbuscular. Arbuscles are, if you look up that word, it means dwarf tree. So here's an arbuscle in the lower right under the electron microscope, and it does look like a dwarf tree. All right, very much like a dwarf tree. And so again, though, that's all just fungal hyphae. So that's where they get the name from that. What I need to point out, and I'll make another point of this in the next slide set, it's really hard when you look under the microscope or the electron microscope. It looks like these fungal hyphae, looks like they're just in the root cells, like swimming around in the cytoplasm, but they're not. They push in, but they're always surrounded by what we call in this case, periarbuscular membrane. So if you look at, let me blow this up a little bit. I'm going to have to do this. Oh, stop. So if we blow this up, so the fungus is in purple, right? So this is the fungal hyphae, purple and pink is the fungus. Green is the plant, green and pink. And so we see the fungal hyphae has pushed up against the plant cell membrane. And it looks like it's in the root cell, but technically it's always surrounded by a plant-derived membrane. Okay, but it, it has kind of pushed in internally, but remember, it's always surrounded. The plant keeps it in check. There's always a plant-derived membrane designating the boundary, right? And that you can't cross. It won't let the fungus cross that. Now, I'm making a big deal about that because in a lot of the published articles about these VAM fungi, they will phrase it, they'll say the fungi live in the root cells. That's not technically true. They're always surrounded by plant-derived membrane. They're not swimming around in the cytoplasm. If they did that, that would trigger a defense reaction by the plant and kill the fungus. And that's not what happens. They live happily together in this very intimate association, and it's a win-win situation. Okay? So make sure you're clear on that. That's one of my pet peeves when I read all these different articles. They're very sloppy on how they describe this interaction. I'll come back to the mycorrhizal fungi in a minute. I just also wanted to mention that there are some fungal pathogens that are considered emerging. That means they're causing more and more trouble, especially with humans. Pneumocystis used to be called Pneumocystis carinii. They've had to rename it. Girovetii. So this is an ascomycete, and they even thought it was a protist, which is a different beast than a fungus. But they're finding it more and more in people who are immunocompromised and then come down with pneumonia. It can be one of the pathogens causing pneumonia. So pneumonia, when you get pneumonia, there's different types. There's viral pneumonia, bacterial pneumonia, fungal pneumonia. So there's several different groups of microbes that find your lung tissue very satisfying to them. Microsporidia are fungi adjacent. They're fungus-related parasites. They are bizarre organisms. They tend to have very small genomes, which makes them very dependent on their host. So probably the most famous one right now, Encephaloptozoan intestinalis, and that one is showing up in AIDS patients. So again, immunocompromised people. And then this one, Rhodotorula mucilaginosa, is also an opportunistic pathogen. Dr. Sharp and I have done some work with this one because it's emerging and they don't know quite how to deal with it. They have been throwing all kinds of antibiotics at it, and it seems pretty resistant. And so they're very concerned about how this particular pathogen may gain a real foothold, especially in people that are immunocompromised. So Dr. Sharp and I have been working with some students, just doing some tests. I have a lot of Streptomyces bacteria in my frozen stock cultures, and so we've been throwing a lot of those against the yeast to see if it can do anything. So these are antibiotic or cold plate studies where we're testing the ability of the Streptomyces. So here, in the middle of this agar plate, you see this bright blue. This is one of the Streptomyces I work with. So that you spot inoculate the Streptomyces after you spread the lawn of this pathogen across the plate. Let that soak in, and then you spot inoculate the Streptomyces. And then you look for kill growth after incubation. So you can see that the Streptomyces is producing something. Huh. All right, weird. You can see different inhibition zones. Whoops, that one wasn't too good. Around. So this one has a really good one. So again, there's promise. Some of the Streptomyces bacteria, I think, are promising for antibiotic, something that this particular pathogen is sensitive to. So your book also has a table here of emerging fungal and microsporidian pathogens. Some of those that I just talked about on this. So again, it's dicey when you have fungi attacking human tissue. It's challenging to find a way to kill just fungus and not harm the human. So it's a very concerning situation, especially if you're already immunocompromised. On a positive note, anyone seen this book or the movie, Fantastic Fungi? Very cool documentary. I don't know if it's showing up on Netflix or anything, I saw it when it first came out. I think it was during the COVID era when it was first being shown. And then they have a book that goes with it, which is the classic, what do they call it? The coffee table book. You know, it's so beautiful. It's good for display. So fantastic images in there, lots of very interesting information, very well written. So if you have an interest in fungi, I would recommend that book. And if you can see the documentary that with the same title, really good documentary. So Paul Stamets is part of that, both the book and the documentary. And have you heard of Paul Stamets? Anyone who's into fungi? He's like the fungal guru here in the United States. And he is pushing very hard for decades, trying to get the word out of how amazing fungi are and how useful they can be. Even as a TED Talk video on YouTube called How Fungi Are Going to Save the World or something like that. And he believes it. So he is a very good advocate for fungi. Okay, and is the second slide set open in EduCat? All right, let me get in there. Thought I had it in there, but maybe not. Ah, there it is. Okay. So if you refresh, it should be in there. So this is part 2 I wanted to get into a little bit. So this slide set, I'm still continuing on with fungi, but I want to just point out a few ways in which fungi are really impacting us or our environment. And I think all mushrooms are edible once. That's true. Here's a lovely poem you can read when you need a poem to read. Very nice. All about mushrooms. You know, that's pretty hard to find a poem about mushrooms, but I finally hit upon that one. Got Mold. Do you remember the Got Milk campaign? So this is a tongue-in-cheek on that. Got Mold. So that's one of the workers in the greenhouse. I had to take a picture of that. That's very, very funny. All right. So huge service that fungi provide to what's going on Earth is cycling, carbon cycling, and other nutrient cycling that needs to keep getting cycled around, through processes on Earth here. We're going to have Earth continue to work as it does. Carbon cycling is a hot topic right now. What do we do with all this, carbon dioxide? Can we get it into a fixed state so it's not impacting our atmosphere? And so carbon cycling is a big thing right now. And terrestrial carbon cycling, how can we use fungi, mycorrhizal fungi in particular, are getting looked at. And they're saying, you know what, up to 70% of the total carbon can get sequestered through the action of mycorrhizal fungi. And so this idea of if we can get the carbon dioxide into the soil, into photosynthesis, that's one way to keep it from, increasing in the atmosphere. And so I just wanted to point out, I'm not going to get into huge detail, but carbon cycling for sure, especially with the mycorrhizal fungi. And this, again, I'm not going to get into detail. This is a visual of carbon cycling on the left here, explanations. Some of this should look familiar. They talk about fermentation, lithotrophy, respiration, photosynthesis. So this is putting it all into context, all that physiology that you learned, putting it into context of carbon cycling and how all those different activities and pathways really, allow for carbon cycling on this Earth. And we got to have it or things are going to grind to a halt. And so again, they're looking at mycorrhizal fungi in the soil. Microbes in general are very important, in particular mycorrhizal fungi. And so there's lots of debate about this idea of, how in the world did you end up with plants on terrestrial environments? So originally, the idea is that plants evolved from green algae. That's the kind of the going thought right now. So in the oceans. So how in the world did you make a jump from totally aquatic to totally terrestrial and survive that when you have no roots? Initially you got no roots. How are you going to do that? And so here they're looking at this idea that mycorrhizal fungi were helping that transition. And here, this particular paper is like, okay, well, what kind of mycorrhizal fungi? So they're starting to debate that, but the evidence is starting to really pile up that this is a thing. They really do think that this is what allowed for this tremendous shift from aquatic to terrestrial, and that they survived that process. And so land plants diverged from aquatic algae in the Neopolozoic as a lineage that would eventually undergo the ecological transition to terrestrial life. This transition, a major turning point in the history of life on Earth, reshaped global climate, biosphere, as roots only evolved after transition to land. How did this happen? And so, how did they survive? And they are pointing to the mycorrhizal fungi. Which group of mycorrhizal fungi it's up for grabs right now. Some people, there's two different camps, I guess, of who's thinking that. Some think the mucormycetes, others think homeromycetes. So that's still up in the air. There's evidence really. The mucormycetes actually are coming into, it could be very much a plausible way. But anyways, just to let you know, it's still up in the air. But they do seem to at least agree that mycorrhizal fungi, especially the VAM type fungi, that's what we're talking about here in both of these groups. And so basically what those slides were just showing, I'm summarizing it here. The green algae or the charophytes, which used to be along the green algae, and now they've separated them out into charophytes. They were the ancestors to the terrestrial land plants, and that's a huge shift. And so if 500 million years ago, if you're going to do that successfully with no roots, the thought is that they must have been able to quickly form a relationship with these mycorrhizal fungi. And the mycorrhizal fungi essentially could feed those plants enough nutrition for them to gain a foothold and start to evolve and adapt to land environment. And so this is a paper, the title of a paper that was published a bit ago. Presence of 3 mycorrhizal genes in the common ancestors of land plants suggests a key role for mycorrhiza. So what they're saying is mycorrhiza in the colonization of land by plants. So they looked at algae in the green algae group, and they looked at their DNA, and they have genes necessary to form a mycorrhizal relationship. So that's really pointing to the idea that the algae were the ancestors to terrestrial plants, and they already had the ability to quickly form a relationship with these mycorrhizal fungi. So it's really cool studies going on with this. The debate still goes on. Is it Mytina? But basically, it's VAM fungi, either group or VAM fungi. So really interesting to see this partnership develop right away that early, 500 million years ago, in order to get them to successfully colonize land. And so when they look at all the different plants, there's a lot of different groups of plants out there. About 85%, some people say 90% of plants have this relationship, some type of mycorrhizal relationship with these mycorrhizal fungi. By far, the majority of plants hang out with the VAM type fungi. Okay, those fungi, and those are the ones that are thought to have helped 500 million years ago. And then about 10% hang out with, the orchids hang out with orchid mycorrhizae. It's a very specialized relationship. 2% are ectomycorrhizae. So much smaller ectomycorrhizae amount in terms of the total. And then there's another group called ericoid mycorrhizae. And so there's different flavors of mycorrhizal fungi, but the VAM fungi, arbuscular mycorrhizal fungi, definitely win, and those are the ones they think were the helpers 500 million years ago. So this is what I mean. So this table is taken from a publication, and they are just looking at 3 different groups. The ectomycorrhizal fungi on the left column, arbuscular in the middle, ericoid on the right. And again, if you look at how they worded it in a scientific published paper, right here, arbuscles and vesicles in root cortical cell. So again, that's a very sloppy way to phrase it because they are not swimming around in the cytoplasm, right? They're always protected, surrounded by a plant-derived membrane. Okay, so even in scientific papers, they kind of mess it up. But anyways, here are 3 different categories of mycorrhizal fungi. Just giving some basic information overview, I guess. And so I'd like to talk a little bit about, especially the next few slides, I'm going to look at ectomycorrhizae and then the arbuscular type. And so this is a good summary table when you're studying, okay? So let me just give you an introduction. So here you're looking at a root in longitudinal section, and they're trying to compare on one image here the arbuscular mycorrhizae and the ectomycorrhizae. And so there's some key differences between these 2 groups. The ectomycorrhizae, what the artist did here is they drew this very thick growth of fungal hyphae, this dark black, on the surface of the root. And so what they're trying to show is that the hyphae grow very tight together. And so when you eat a mushroom, essentially what you're eating is, fungal hyphae that's really tightly compressed into the mushroom design. Here you have very tight hyphal growth, very tightly compressed over the surface of the root. So that's called the mantle. I'd like you to know that word, please, mantle. Okay, so when someone says, oh, look at that mantle, they're referring to fungal tissue growing on the surface. Very thick. So ectomycorrhizal fungi can do that, and that can be very protective. The root's now physically protective by this very thick growth. And so it's harder for something to come along and eat at the root because the fungal hyphae are covering it. The other term I want you to know is Hartig net. So the Hartig net is this idea named after Dr. Hartig. Okay, she came up with that. He looked at this under the microscope and realized that the ectomycorrhizal fungi are growing in between the roots cells, right? So they're just hanging out in the cell wall space in between the cells, all right? And so it looks like a net. If you got, it looks like a fishing net when you look at it under the microscope. All the fungal hyphae, the way they weave around all the cells, it looks like a fishing net. So they named it Hartig net in honor of Dr. Hartig. And then the other idea I want you to capture here is that you see these fungal hyphae growing off the root and into the soil. That's where the big payoff is for the plant. If it can form this partnership with this fungus, and then the fungus grows its hyphae way off into the soil, and it can be super extensive. And now that fungal hyphae can absorb water, absorb minerals, and it will feed a lot of that back to the plant. And so, and the other thing is fungal hyphae are very small in diameter compared to even the finest root. And so the fungal hyphae can get into tiny crevices of the soil that the root just physically can't do. And so when you have this relationship, the plants benefit tremendously. Uptake in water is increased for sure. Key minerals, in particular phosphorus, is hugely increased. Plants have a hard time getting access to phosphorus. And mycorrhizal fungi are like, hey, I can do that. And so it's a very good partnership. On the left side of this diagram showing how the arbuscular mycorrhizae interact with the root, you don't see a mantle, no mantle. Now there is some growth along the surface, but it's very light and delicate. So if you were to dig up the roots, it would get stripped away. You wouldn't notice it because it's so light and delicate. You do see the hyphae coming off again. They do the same thing. They'll grow their hyphae off the root into the soil and get access to all that water and minerals that the plant needs. And then you see the arbuscles and vesicles. So what they do, we say it's more intimate compared to ectos because the arbuscular, they can put their hyphae into the cell wall spaces in between all the cells. they're doing that, but then they can push up against the cell membrane, so they can actually get through the cell wall, bump up to the cell membrane, and kind of push in a little bit, but they're always surrounded by the cell membrane of these root cells. And so that's a little bit more intimate because now you have fungal hyphae and cell membrane making contact rather than just cell wall. Right, so that it's a little bit more intimate, and it's good. It's a good way to exchange chemicals back and forth when you have your membranes bumping up to each other. Right. All right. And you've already heard about the vesicles and arbuscles terminology. So let's take a look here. If you were to dig up some roots of trees around here, you could see something like this. So these are roots covered in the mantle. So all of that thickening, the peach color in the top left, that's all fungal hyphae, really tightly growing together and tissue-like growth. And then I put this lower right image. This is more of what you see with the naked eye. You see how small the finer root get covered with the fungal hyphae in this white. And then also they're showing nicely the fungal hyphae coming off the roots into the, you have to imagine that going off into the soil. Here's another image, nice, showing the hyphae growing off, and imagine that going into the soil. And then the endomycorrhizae, you know, they don't have that thick mantle around the root, but they certainly do have the hyphae going off into the soil. They have very special looking designs when you look at how they grow in the root tissue. So very much the plant gets really, it's a win for the plant because they get access to water, extra water. And then I ranked it out, P and K. Phosphorus by far is the best advantage when the plant forms this relationship. The mycorrhizal fungi can get access to phosphorus that the plant just has a really hard time with. And so we see huge difference in phosphorus uptake when the fungus is there compared to if it's not, and also nitrogen and potassium. And probably thousands of studies have been published now on how mycorrhizal fungi can stimulate seedling growth. They protect the plant both physically and chemically with antibiotic production. And then the fungus, if everything's going okay and the plant is nice and healthy, the plant, of course, is doing a bunch of photosynthesis and it's able to deliver sugar to fungus. And some of the studies that they've done in forested systems out in British Columbia, they show 30 to 40% of the sugars the plant makes get sent to the fungus. And so if you're going to devote that much of your sugars to your partner, that's saying you want that partner to stay with you and to continue to live with you. And then they also get some other chemicals, growth factors in general, that helps the fungus. So this is considered definitely a win-win. And you can certainly, like I said, you can buy mixtures of, especially the BAM type mycorrhizal fungi. You can buy those off Amazon and other places. People are really starting to grow those up. There's been more work doing with cannabis research, looking at that relationship. Now that it's legal to start do more research on them, they're looking at the mycorrhizal relationships and how that might influence the secondary metabolite profile, whether it be CBD or THC. Looking at that, and they can see a difference for sure, presence or absence of the mycorrhizal fungi. So there are some cannabis growers. I'm on a committee for one of the grad students here that is looking at different organic amendments, living soil type things, and looking at the impact of that on his cannabis plants. So lots of interesting work looking at this partnership. Orchids have their own little suite of mycorrhizal fungi. Orchids are amazing plants. Unfortunately, we don't have a ton of them around here. You have to go into the tropics. This is Ram's Head Lady Slipper. And my former grad student, Carrie Farkas-Lasich, who now teaches in the indoor ag or controlled environment program, she did her whole master's thesis on this orchid and some of the endophytes that coexist with this particular orchid. But orchids in general, their seeds are like particles of dust. They're so tiny. They have no nutrient reserves in the seed. I don't understand why orchids did that. Seems like a poor strategy. The only way orchid seeds germinate is if they have the right mycorrhizal fungal partner right there, wherever that seed landed. I mean, what are the odds? And so this is really kind of puzzling to me, but that's how they work. Tiny dust-like seeds float away. Hopefully there's the correct mycorrhizal fungus there, and they have to have an interaction for that seed to germinate, and then the fungus has to feed that baby orchid. Otherwise, the orchid dies. It's an amazing relationship. And so 0.3 micrograms, that's their weight. I mean, it's just incredible. It's literally dust-like particles. And so they are totally dependent on that fungus for quite some time. And they are, so this is a very special dynamic, and there's very specific fungi that need to partner with a specific orchid. So it's not just any old mycorrhizal fungus that can do this. And again, so it just blows my mind that orchids are so abundant when you have such picky parameters here in order to get those seeds to germinate. So super specific association between the fungal has to be a very particular fungal species to that particular orchid species. And otherwise, the seed won't, the fungus tends to use it and essentially eat it up for its own nutrition. It won't form a partnership. It's the wrong fungal species. So orchids are really, they're a huge group of plants. They're very, very successful. There must be a lot of these types of mycorrhizal fungi in the tropics because that's where most of these orchids hang out. So they look interesting under the microscope. They have these fungal hyphae that they have called pelotons, and they're coiled hyphae that exist. Again, they're surrounded by membrane, but they show up in the orchid root tissue, and then just this continual. The fungus continues to grow. Some of the hyphae die as more grows, and so it's a very dynamic interaction. And so orchid, for the longest time, people didn't know how to grow orchids, and I think it was back in the Victorian era of our history that people would be employed by rich people to go hunt orchids. So they were called orchid hunters, and they traveled the world to go look for rare orchids and rip them out of their habitat and bring them back to these rich people so that they could put them in their own private collection. And there's some books, at least one book that I read about this, where people were like fighting over orchids. There was even murder. People murdered each other. One person was murdered because they had some orchids the other one wanted. So, I mean, it's just crazy. It was kind of ridiculous. But they were doing all that because they didn't know how to grow these orchids. They didn't know that they needed this mycorrhizal fungus. So they had access to all the seeds they could want, but they had no idea how to make them germinate. So they went and hunted them instead. So, all right. The next topic, I just have a few more things to say, but I'll save that since we're running out of time. We'll talk about more of how mycorrhizal fungi are impacting forested systems and some other fungal topics. All right. And then we'll get into the next topic on Friday as well. Viruses. We're getting into viruses.